Alright, welcome to part 2 of cell reproduction notes. Once again, this cell right here is not in cell reproduction. The reason we know that is because the nuclear membrane is still intact in this cell and we do not see any chromosomes yet. You cannot visibly see the chromosomes under a microscope. It would look something like that if it had started cell division. Same thing with this guy over here. It's just a drawing of it. Remember these two red structures? Those are called the centrioles. They're going to begin migrating to opposite ends of the cell once we begin mitosis. But we do not see any chromosomes in here. We do not see any chromosomes. Therefore, we know it is in interphase. This cell is not dividing yet. Alright, so the first stage of mitosis, remember we had our little word PMAT to help us remember the order of the different phases, and PMAT starts with a P. So our first stage of mitosis is going to be prophase. This is where we can see down here, this is a cell. We cannot see any chromosomes inside that nucleus. This one is still in interphase. This is a plant cell who has entered prophase. And this is an animal cell that has entered prophase. We no longer see the nuclear membrane around those chromosomes. Now we can clearly see these dark little worm-like structures in there. Those are the chromosomes of that plant cell. Same thing with this animal cell over here. It no longer looks like a clear, like pink color, like over there in interphase. You can clearly see the dark rods, the chromosomes. They're not arranged in any pattern yet. They're just kind of in a big ball. And that is basically all you need to know about prophase. The nuclear membrane disappears, and the chromosomes become visible, but they're not arranged in any pattern yet. That's prophase, the first step. And pretty much the same thing, just in like diagram form. Here's the nuclear membrane. It is going to dissolve. Here's our centrioles. These are the guys that are going to pull the chromosomes apart. They're going to move to opposite ends of the cell. This little red line right there, those are the ropes that these spindle fibers are going to use to pull the chromosomes apart. And those are called spindle fibers. So those red little ropes right there made of proteins, those are called spindle fibers. Once again, you can see this cell down here. It is in prophase. We can clearly see the dark chromosomes. There's no longer a nuclear membrane holding those chromosomes in. Nuclear membrane has dissolved, and we can see the chromosomes. Alright, the second phase in PMAT. We already did prophase, so now we're up to the M phase, or the metaphase. This is an easy one. We can still see the chromosomes right there in that animal cell, right here in that plant cell. All you need to know in metaphase is that the chromosomes are no longer in like a ball. They're no longer in just a big old clump of chromosomes. They've now become arranged. The chromosomes are lined up in the middle of the cell in a place called the equatorial or the metaphase plate. But all I care is that you can recognize that these chromosomes are now arranged in a single file line right along the middle of the cell. Same with these guys right here. They're no longer in a big blob of chromosomes. They're lined up straight in a line right in the middle. These would be the centrioles right here and right there. And these are the ropes. You can barely see them. They're real faint. But those are the spindle fibers that are going to attach to each of these chromosomes. And in the next phase, they're going to pull the chromosomes apart to opposite ends of the cell. Same thing over here with the plant cell. You can see the spindle fibers right there. Can barely see them. But these chromosomes are in the next phase, they're going to get pulled away from each other. But the main thing, metaphase, just think middle. The chromosomes are lined up in the middle during metaphase. Alright, so once again, here is metaphase in like a diagram. You can see they're all lined up in the middle. M is for metaphase. And that just means the chromosomes are all lined up in the middle. Chromosomes are in the middle, that means metaphase. Now remember, this red chromosome, it has two sister chromatids. Each one of those legs, in the red guy, the green guy, the blue guy, and the yellow guy, these are exact copies. So this green leg is the exact same thing as that. This blue leg sister chromatid is the same exact copy as that one right there. And in the next stage, these sister chromatids are going to get ripped apart from each other, and they're going to get dragged to opposite ends of the cell. Here it is again down here in this real animal cell. You can see that the chromosomes are all lined up in the middle, and that means metaphase. Alright, so PMAT. We've done prophase, the P. We did metaphase, lined up in the middle, M. Now we're on the A, or anaphase. This is when the sister chromatids are being pulled away or apart from each other. So, in this plant cell right here, each of these chromosomes are getting pulled this way, these chromosomes are getting pulled that way. While they are in motion, while they're getting pulled to opposite ends of the cell, that is anaphase. So whenever they're moving away from each other, that equals anaphase. Same thing down here with this plant cell. This is early anaphase. These chromosomes have barely started to move apart from each other, but this group is moving that way, and this group is moving that way. Anaphase, all you need to know is that the chromosomes are moving away from each other towards opposite ends of the cell. Alright, anaphase in drawing form. So this red sister, these used to be together, they used to be stuck together at the centromere, right there. They used to be stuck together at the centromere. Now that they've been ripped apart, they're now single-stranded chromosomes. They're no longer chromatids. So these two reds are exact copies. Same with these two greens. This green guy's going to go over there. This green guy's going that way. This blue guy's going this way. That blue guy's going that way. Same with the yellows. And remember, each one of these chromosomes are exact copies of each other. So when they get to the end over here, each new cell they're all going to have a red one, a green one, a blue one, and a yellow one. So these two cells are going to be exact identical copies of the original cell. Alright, the last one of our PMAT. Prophase. The chromosomes became visible and the nuclear membrane dissolved. We did that. Metaphase. The chromosomes all lined up in the middle. We did that. Anaphase, the A. The chromosomes were moving away from each other or apart. Did that. Now we just got the telophase. This is where the chromosomes have arrived at opposite ends of the cell. So the chromosomes are no longer moving away from each other. They've arrived at opposite ends of the cell. A new nuclear membrane begins to form around each cluster of chromosomes. You can see that here in this plant cell, and kind of see that there in this animal cell over here. And the final thing that needs to happen before these two cells can divide is a process called cytokinesis. So in mitosis, we made two nuclei. Nucleus contains your DNA. Well, in mitosis, we made two of them. But now, these two nuclei are still in the same cell. So we need to break this cell off and form two cells. Same thing over here. We got two nuclei in the same cell. Well, this cell membrane, the cytoplasm, is going to pinch off. And when it does, it's going to form two cells. And this process of the cytoplasm pinching off to form two new cells, that's called cytokinesis. And here it is again, a little bit better pictures. So the two groups of chromosomes have arrived at opposite ends of the cell. A new nuclear membrane is beginning to form around these chromosomes. You can see that this guy over here and this one over here, they're going to be exact copies of each other because they all have the same type and the same amount of chromosomes. You can see that the cell membrane here is beginning to squeeze in. Well, this squeezing in is called cleavage. And when it totally squeezes together and pinches off, that's called cytokinesis. All right, so this is telophase at the end. New nuclear membrane is surrounding the chromosomes, and the cell membrane is going to begin to pinch off to form two new cells. All right, so here's a picture of the entire cell cycle. It's not really to scale, but this whole side of the circle, all the way from the green all the way over here to the blue, that whole, 3/4 of it, that's interphase. So all of this is all interphase. So over 90% of the time, cells are in interphase. And remember, this is the G1 where the cell is growing. This is the S phase where the DNA is being copied or replicated. And then this would be the G2 phase where important organelles are being made, preparing the cell to divide. Here is the little mitosis section. So this is the time when a cell is dividing. It's in cell division. And there's our PMAT, prophase, metaphase, anaphase, telophase. And of course, at the end of cell division is that process called cytokinesis. So remember, the vast majority of the time, cells are in interphase. A very small percent of the time are they dividing? Are they in mitosis or cytokinesis?